Okay, good afternoon, everyone. And today we're going to begin a very important section of work. And this is to discuss microbial genetics, particularly from the point of view of the functioning of DNA in the prokaryote cell. So just to remind you of a little bit of background already, we have already discussed the structure of DNA and of the nucleic acids. I will review that for you at the start of this class again. But please make sure that you go back and that you carefully go through all of what we discussed when we talked about biomolecules, about the structuring of the nucleic acids, DNA and RNA. Second, remember that in the prokaryotes, the major part of hereditary material is lodged in a single bacterial chromosome. It is a circular chromosome. It's attached to the plasma membrane, the cell membrane, at one point called the origin, usually designated ORI. And so I say most of the hereditary material of a prokaryote cell is in this large circular chromosome. But there are other genetic elements as well. And these are small circles of DNA called the plasmids, which are independent of the chromosome and often are acquired from the environment. They originate in other bacteria, but plasmids are essentially infectious. They can be taken up by bacterial cells, and they often contain very useful information for the bacterial cell. That large chromosome has to replicate every time the cell divides. It replicates, and as far as possible, that replication has to be very, very accurate, because two daughter cells have to inherit exactly the same genetic information. The plasmids also divide. They may divide, it depends on the structure of the plasmid. Some plasmids divide when the rest of the DNA divides, when the chromosome divides. Others divide independently. It just depends on the chromosome. There are different mechanisms for plasmids to reproduce inside the cell. So that's where most of the DNA is going to lie that we are going to talk about. We're going to talk mainly about the bacterial chromosome. And then we will talk a little bit about plasmids in another class when we start to talk about genetic engineering. Okay, so let's have a bit of a review first of all. This slide you have seen before. It forms part of our discussion about biomolecules. And so when you want to revise this structure of the nucleic acids, use this slide because it contains virtually all of the information that you need. For a start, the nucleic acids are polymers. They are often very, very large molecules. DNA is extremely large. Even in the bacterial cell, where there's just a single chromosome, that chromosome can be very, very, very long. And we need to understand how that polymer is actually made up. It's made up, the monomers that make up the polymer are nucleotides. And here is a single nucleotide, this whole unit here. Without this part here, we refer to as the nucleoside, but the whole thing together is a nucleotide. At the center of the nucleotide, there is a sugar here. There are two sugars that are involved in the nucleic acids. We'll wait a minute and we'll talk about them. So this here is a 5-carbon sugar, 1, 2, and let's see where the carbons are. That's 1, 2, 3, 4, 5. So 4 participate in this ring structure, and the 5th is branched off here in this group here. So it's really important that you understand that how these are named. They are named by their position. So this here is the 1' carbon, 2' carbon, 3' carbon, 4' carbon, 5' carbon. They just aren't shown here, just for shorthand's sake. There'll be a C there, C there, C there, C there, C there. 1', 2', 3', 4', 5'. We need to remember the naming of those sites, because they become very important in our understanding of how our DNA actually functions. At the 1' site, there is a nitrogenous base of some sort. There are, in fact, 5 bases that participate in structuring nucleotides for the nucleic acids. So this could be any one of 5. And here they are: cytosine, thymine, uracil, adenine, and guanine. They can be located there. Some you see are 1-ringed, some are 2-ringed. We'll discuss that in a second. At the 5' site here, there is a phosphate group. In actual fact, when the nucleotide is free in the cell, there's actually usually 3 phosphate groups attached here. When the nucleotide is located in a nucleic acid, there's just 1, for reasons that we'll see in a while. So the next thing to realize, there are 2 kinds of sugar that can be involved. The first is a ribose sugar. Ribose sugars have a hydroxyl group at the 2' site. Here's the 2' site. Ribose, they would have an OH group, a hydroxyl group there. That's ribose. The other sugar that we will see is deoxyribose. And deoxyribose will take off that oxygen there, and you just have an H there, a hydrogen. And that is deoxyribose sugar. So that should immediately spark something in your brain, because we have 2 kinds of nucleic acid. The first kind is ribonucleic acid, and it gets its name from the fact that it utilizes ribose, not deoxyribose. DNA is deoxyribonucleic acid, and DNA uses this. It uses the deoxyribose sugar as the skeleton for the nucleotide. Okay, so we can tell, and the cell can tell immediately what the difference is between 2. The cell sees RNA, it recognizes it because it sees this, amongst 1 or 2 other characteristics, but it sees this, and the cell recognizes that this is RNA that it's working with. This here, it sees that, and it knows, the cell will know enzymatically, it will know this is deoxyribonucleic acid. So that's the importance of the different sugars. We get 5 different bases, 2 different sugars. Now, all of the nucleotides, when they're free in the cell, they have 3 phosphate groups attached. If I want to make the polymer, the polymeric nucleic acid, then I can add nucleotides to an already existing chain. But there's a very, very strict rule that you must remember. When you go to bed at night, you lie on your pillow, you say 3' hydroxyl, 3' hydroxyl. To remember that this here, this site here, 1, 2, 3, the 3' hydroxyl in the nucleic acids, no matter what nucleic acid you're talking about, this is the only place which is reactive on the nucleotide. It's the only place where you can add another nucleotide. So if looking at, here's a chain of nucleotides, in this case, just a single chain. There is the 3' hydro. This is our last nucleotide. 1, 2, 3. That's the 3' site. That's the only place where we can add another nucleotide. Let me take this nucleotide and add it here. And what I'll do is I'll end up making a very strong covalent bond between this phosphate group here and the hydroxyl end there. So what we see when we look at the polymer, what we see is a backbone, and it's alternating sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar. If I wanted to add another nucleotide, I have to bring the nucleotide in and add it there, and I add it via the phosphate groups. We'll see in a while how it's done. But I'll end up putting another one. I cannot add on there. I cannot add on at the 5' end. It won't work. Instead, I have to add here. So let's just remind ourselves also that DNA is a double helix. It consists of 2 of these backbones, and they are united across the middle because these bases here, these bases are structured with lots of nitrogen, and they very, very easily make hydrogen bonds. And they make hydrogen bonds across the middle. In DNA, there are 2 strands, and they'll make hydrogen bonds across the middle. And there are always 3 rings across. Remember here, we have some of our bases had 2 rings, others had 1. There are always 3 rings across the middle in DNA. And DNA achieves that by always matching cytosine to guanine, 1, 2, 3, adenine, 1, 2, to thymine, 3. In DNA only utilizes cytosine, thymine, adenine, and guanine. And it does it because it matches, it forms hydrogen bonds between guanine and cytosine, between adenine and thymine, to link the 2 backbones together. There's another feature of another consequence to this only being the only reactive site, which we'll see in a minute that we need to pay attention to. So these are the nitrogenous bases. Anything with 1 ring here, we call a pyrimidine. And so our 3 pyrimidines are cytosine, uracil, and thymine. Uracil is a base which is only found in RNA, and it replaces thymine. Thymine does not occur in RNA. So uracil, the presence of uracil is another label to say this is RNA. Look at the structure here, and you'll see they're very, 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 very similar to one another. Thymine has this methyl group here, which uracil doesn't. But otherwise, they're very, very, very similar. But the presence of uracil in a nucleotide labels it as RNA. And you just remember that our thymine always linked to adenine. Well, whenever uracil has to link to another base, which it sometimes does, it will link to adenine, as if just the same as thymine would. The 2-ringed structures are purines, and our 2 purines are adenine and guanine. So that's all very confusing. You've got to kind of know, oh, it's a 1 ring, oh, it's a 2 ring, pyrimidine. So I'm going to tell you a little story. And I'll just do this very briefly. I usually spin this out with great drama in class, but I'll just tell you this little piece of ancient history. And it is to tell you that in ancient Egypt, there was a pharaoh. All his ancestors built these huge pyramids, like in the background. And he didn't want to. He wanted to, instead of building that great big pyramid, he wanted to build a very cute little pyramid. It was very beautiful. This was a great departure, and in order to even use the pyramid, he had to get the priests, the temple priests, to come and agree to purify it before it could be used. And everybody said, no, you're not going to, it's too different. But he did anyway, and he went and got the priests. They took a look at it. They loved his little pyramid. And they agreed to purify one cute pyramid. And there it is there. Agreed to purify one cute pyramid. A and G, adenine and guanine, are two-ringed purines. Adenine, guanine are two-ringed purines. Basically, that's all you need to know, because everything else is going to be one-ringed and a pyrimidine. But agreed to purify one-ringed structures are cytosine, uracil, and thymine. CUT, cytosine, uracil, thymine, one cute, and they are pyrimidines. Okay, agreed to purify one cute pyramid. Just remember that, and you'll be able to remember exactly what's one ring, what's two ring, what's a purine, what's a pyrimidine, with their names. No excuses. All right, so what we said in DNA is two-stranded, right? And when you have when you have A on the one strand, it will always match to T on the other strand. If you have C on the one strand, it will always match to G on the other strand. So if I have one complete strand of DNA, I always can complete the matching strand very easily, simply by putting in the matching bases. So let's have a look at this one. And this is A 5 prime ATTAGCC. What is this here? Just have a look back here, and you'll see that if we look at the polymer, and there's lots of nucleotides joined together, because this 3 prime site is the only reactive site, this end here has a free 5 prime end there. It's a free 5 prime end. At the other end here, there is a 3 prime site. So we actually label the strand 5 prime to 3 prime. It's very important that you put the priming in, that you have the orientation of the strand, because the opposing strand comes in the opposite orientation. We will see that this strand here has its 5 prime end here and the 3 prime end here. We say that the two strands are anti-parallel. They are parallel, but they are anti-parallel. Their orientation is exactly opposite. 5 prime, 3 prime. The matching strand would be 3 prime, 5 prime. Okay. So it's very important that we put in the priming in this little strand here. I'm asking you, if you know one strand, you must be able to do the other. What do we have? 5 prime ATTAGCC. What is the matching strand going to be? Shout it out. Shout it at your computer. 3 prime. Okay. What is the matching base to A? Thymine, T. What's the matching base to T? Adenine, A. What's the matching base? A. What is the matching base? T. What is the matching base? Guanine to cytosine, C. Cytosine. What is the matching base to cytosine? Guanine. Guanine. So I hope that I did it correctly. TAATCGG. That is the matching strand. And notice the orientation. This one is 5 prime ATTAGCC. The matching strand must be 3 prime TAATCGG. Here's a longer one for you to do. Take a minute. Work this out for yourselves. What is the matching strand? Write it down. Right. It is 5' CC G T C G C A T T T T. Okay. What is the priming at the end here? For this strand, 3'. For this strand here, 5'. Okay. Remember, they match A to T, C to G, and they are anti-parallel. So it's 3' on the one strand must be matched by 5' on the other strand. This, we labor this point because this not only tells us the structure of DNA and how DNA is put together, it also gives us the mechanisms whereby DNA is actually used. How DNA, for example, replicates is very closely tied to this fact that if we have the one strand, we can reconstruct the other strand very easily. If we look at this double DNA strand here, and we'll notice all of the things that we've already talked about. First of all, here is the one strand. Here are the sugars, the deoxyribose sugars. How do I know they're deoxyribose? Because this is a double strand, and it's got thymine in it. That means it is DNA, therefore this must be deoxyribose sugar. All of the deoxyribose sugars are joined to one another by these bonds with the phosphates all the way down. Very, very strong bonds. This molecule is very, very strong, very resilient. At the 1' site on each of these deoxyribose sugars, there is a base. In this case, the bases are T, G, C, A. But there's something very important that you need to note. Just because I've got A here does not predict what the next base is. I can bring any of the 4 bases used in DNA. I can bring any of them in here. This is important because it means that the order of bases is infinite. There's an infinite number of orders of bases. The order of the bases is not random. It's very, very strictly guarded. We say it's strictly conserved by the cell because locked into the sequence A, T, C, G, T, whatever, locked into that sequence is the actual functioning of the DNA. Locked into that sequence is a code. And that code is for the proteins that the cell requires. So all of the recipes for the proteins the cell requires are locked into the DNA, and they're locked in the form of the sequence here of bases. It's just that there's an immense, infinite variety of orders. With that simple basic structure of 4 different nucleotides carrying the bases adenine, guanine, cytosine, and thymine, with just those 4 nucleotides, we can have an enormous amount of different information present in the DNA itself. So first of all, sugars, deoxyribose, joined with phosphate groups, joined with these very strong phosphate bonding. These are called phosphodiester bonds. Across the middle, each nucleotide has its own individual base. And across on the other side, the matching strand has to match this base sequence, A to T, C to G. Here it is here. But in order to do so, the matching strand is anti-parallel. Have a look here. Here's the 3' end of this, and here is the 5' end of this. You can only add bases at the 3, all activity with nucleic acids takes place at the 3' end. If I want to grow the strands, I could only add here. I can only add here. So when we start making a DNA strand, when we start linking bases together, start linking nucleotides together, we can only add them here. So when we watch the growth of an individual strand, this strand will grow in this direction, that direction. This strand would grow in that direction. So whenever we add bases and watch a strand growing, as we will in a little while, we only see the strand growing 5' to 3' direction. 5' to 3' direction. Because you can only add here. You can only add at the 3' end of each of these strands. The strands are anti-parallel and they are match across the middle. You'll also I'll just point out one thing. These little dotted lines here tell you how many hydrogen bonds can be made. And first of all, we are always matching T to A, purine to pyrimidine, right? T to A bonds with 2 hydrogen bonds. G to C, again, purine pyrimidine, always match across the middle, and we'd have 3 bonds there. All right. So that gives us our orientation now. This is shown as lying flat here, just for convenience sake, we flattened it against the page. But remember that the DNA molecule, the double molecule of DNA, is this beautiful lazy spiral, a double helix. It has a very distinctive 3-dimensional shape in space of a double helix. Okay, here's the very, just let me flip forward here. So what is the responsibility of DNA? The responsibility of DNA is to carry all of the information necessary for making all of the proteins in the cell. And that most importantly means that DNA contains the recipe for the vast majority of the protein enzymes that are driving all of those processes of metabolism, growth, movement, et cetera, et cetera, et cetera. All of that is lodged in the DNA, and it's contained in the code of the DNA nucleotides with their attendant bases. So the first thing is that the hereditary material contains this vital information. And when a cell divides, it's important that that information be handed on entire and correctly to the 2 daughter cells, so both of them function as well as the original cell. And in order to do so, the cell has got to reproduce the DNA completely before the cell divides. And it does so in a process called DNA replication. And the process of DNA replication at its base is extremely simple, because we've already learned that if we take away one strand, if I obliterate this strand, take it away, I can recreate it as long as I have the other strand complete, because A must always be matched to a T, C must always be matched to a G, G must always be matched to a C, etc. If I take DNA and I just have the one single strand, I can automatically create the other strand as long as the one strand is complete. So we say that each strand serves as a template. It serves as a template for the production of the second strand, and that applies to both strands. So here's the way in which this replication takes place. First of all, the replication of DNA is performed by a whole big complex of enzymes. There are numerous enzymes involved in DNA replication. And we put them all together, we call it the replication factory. So in prokaryotes, when the cell needs to divide and needs to replicate the DNA, cellular messengers are sent to enzymes, et cetera, and the replication factory assembles and begins to perform the replication at that place where the DNA chromosome is attached to the cell membrane at the origin, O-R-I, and it will begin there. We're just going to follow one side. It actually happens on two sides, but we're going to follow just one side. Here's the replication. The first thing it does is it opens up the DNA. It splits those hydrogen bonds apart. So have a look here. Here is the DNA, the normal DNA. And here it is enzymatically split those two strands apart from one another. That's actually quite easy to do. Those hydrogen bonds are relatively weak, not like the covalent backbone. They're fairly easily separated. There is a problem though, and it is that the cell does not like to see single-stranded DNA. When a cell sees single-stranded DNA, it thinks virus, because viruses quite often have single-stranded DNA as their genetic material. It's very strange. We'll talk about it when we talk about viruses. If the cell sees single-stranded DNA, it will attack it and try to chop it up into little pieces to render it inactive. So immediately these strands are separated from one another. They get stabilized by proteins which adhere to the strands, and they keep the two strands apart. First of all, they stop it from just joining together, but they protect the strand from enzyme action by the cell. So those are stabilizing proteins. So this opening up here is performed by an enzyme called helicase. There it is there, and what the helicase is, it effectively is moving down the strand, unwinding the DNA and opening it up. And this we call the replication fork there. And it's important to realize this is moving, and it's moving in that direction. It's moving that direction there. Now, just picture this. This is a double helix. It's like a string wound up. Just picture if you had a string and it was attached at one end, and then you fiddle with it, open up the two strands of the string and pull them apart. What happens? Well, you introduce tremendous tension down that you force tension into the downstream part of the string. And one of the things that happens if you do this with a real string, you'll see that one of the ways that has of mechanically relieving that stress is to snap into supercoils further down. It forms additional coils. That relieves the stress, but it's very, very dangerous for DNA, because that's exactly what happens to DNA. If we open it up like this and start running that replication fork down the DNA strand, which is a double helix, we introduce tremendous stress and tension further down. That stress and tension is undone by a very, very specialized enzyme called gyrase, G-Y-R-A-S-E. You'll see its name again in a minute. Gyrase is one of a family of enzymes called the topoisomerases. The topoisomerases are all concerned with maintaining structure of DNA. Because there are several times in functioning DNA where we do this sort of thing. We open it up and we introduce all sorts of stresses and things. The topoisomerases, of which gyrase is one, ensure that the structure of DNA is preserved. They relieve those stresses further down. Okay, so now we've opened up our strand. We've opened it up on both sides. Well, remember what I told you. We can only add bases at a 3' end. There's another feature of DNA replication, though, which is really important, and that is You cannot just simply start adding DNA nucleotides. You cannot just simply start adding deoxyribonucleotides to this. Instead, in DNA replication, a small length of RNA must be laid down first. And this is called the primer. And it's laid down by an enzyme complex called the primase. It's an RNA polymerase. It's RNA polymerase lays down this short primer here at the start where replication has to begin. That RNA primer is exactly the same, it has exactly the same feature as DNA in that you can only add at the 3' hydroxyl site. So let's have a look. This is where the orientation of the strand starts to become very important. You need to know what is the orientation of the strand. In this case, this old strand here there is its 3' site. So its 5' site would be down here. Okay, 3', 5'. Here we're going to lay down a little RNA primer, and we're going to start adding bases to the RNA primer. We're going to start adding now DNA bases. Once we've laid down the primer, then we can start. We're going to lay them down. That's the 5' end of the primer. Here's its 3' end. We can only add at the 3' end. So we're going to be adding in this direction here. As I told you before, we add 5' to 3' direction. So these bases are added here by an enzyme, a very important enzyme called DNA polymerase. So DNA polymerase adds in the deoxyribonucleic acid bases, and it adds them in to match the old strand. Where there's A on the old strand, it will add T on the new strand. Where there's G, it will add C. Where there's C, it will add a G, etc., etc. And what's important to notice here is the growth is occurring in the same direction as we are opening. Remember that this helicase is busy moving down, continually opening up new bases. And this DNA runs behind and just adds in the new bases. Once we've added one primer here, it can carry on for thousands upon thousands of bases without adding another primer. It just carries on in the same direction. Okay, well, that's fine, but we got a problem. And the problem lies in the fact that we can only add at a 3' site on the new strand. And for each new strand, we need a primer to start it. This is a problem because look at this strand here. Its orientation is opposite to this strand. Here's the old strand. There is its priming. It's 5'. So if this is 5' and this is the 3', let's just take this here. We're going to have to add in this direction here. We're going to have to add in the opposite direction to the way the fork is opening. But the problem is that we've got to put down a primer first. So let's have a look down here. Well, I put down a primer, and then I can add in that direction there. But in the meantime, this fork has opened up behind, and these bases now need to be matched. We've got to put down another primer and add in that direction. As we finish that, the fork has opened up even more, and we've got to now add another primer. The impact of this is that for this strand here, which is adding bases in the same direction that the fork is being opened, we form a continuous strand. This strand here, the opposite strand, we have to add bases in the opposite direction to the way the fork is opening, and we end up having to lay them down in short fragments. And those fragments are called Okazaki fragments. There's the name there for you. Okazaki fragments. We call this the lead strand, and we call this the lagging strand. So on the lagging strand, we have to continually lay down these new primers, new primer, new primer, add base, add the base. We end up with these fragments of mixed RNA and DNA. RNA primer, a short length of DNA. Then it bumps into the primer that was already laid down and stops. Then we got the new primer, DNA, primer, DNA, primer, DNA. These Okazaki fragments all the way down here. When as this process proceeds, another DNA ligase, another DNA polymerase comes in, and this DNA polymerase is actually able to go through the primers and chop and digest them out and replace them with DNA nucleotides. So this DNA polymerase will run through here, and it will replace all of the primers with DNA. This leaves a gap in between where the fragments were. Each of the fragments is interrupted by a single little gap, and that gap is repaired by this enzyme here called DNA ligase. So DNA ligase heals up all of the breaks or nicks in this strand and ends up creating the combination of action of this DNA polymerase and the ligase is to create a single strand of DNA. A similar process happens here. We actually need to digest out that RNA primer and replace that with DNA as well. That is done. And again, this is healed up with DNA ligase. But on the lead strand, we lay down thousands upon thousands of bases of DNA before we have to lay down another RNA primer. Not the case on the lagging strand. We have to do it over and over and over and over again. So DNA polymerase creates the new strands. On this one, there is a special DNA polymerase which can chop out the RNA primer and replace it with DNA, and then all the gaps are healed up by DNA ligase. And at the end of all of that process, when we look at what we have, we see that we have the old strand here, a parent strand, and attached to it a new strand. And that new strand should match this faithfully. This is the other old strand, and here is another. We will end up with another new strand there. So after replication, we end up with two identical DNA strands. And on each of them, one half is parental and one half is new. One half has been conserved. So we say that this replication is semi-conservative. It is a semi-conservative replication. Every time we look at the DNA molecule, what we're seeing is one parental strand and one new strand that was laid down during DNA replication. Okay, this is basically just a summary diagram for you to remind you of what I've just said, of the way in which the DNA is made and the DNA is replicated. And what is really important to remember is that the leading strand synthesized in one direction for a very, very, very long time. The lagging strand is laid down in these fragments because we have to continually lay down primer and then lay down DNA in the opposite direction to the way that the fork is opening up. So we recognize, this is also like summary for you. We recognize these three periods during the replication. The first is initiation. Initiation is when the replication factory first binds to the DNA, and it binds at the origin. the full name for the origin is the REC. So the DNA factory, the replication factory binds at the RE and begins to open up the fork. All of the rest of the stage is elongation, and that is the synthesis of the new strand in matching the old strand, first by laying down an RNA primer, and then by laying down all of the DNA nucleotides. On the lagging strand, elongation entails taking out all of the RNA primer, replacing them with deoxyribonucleic nucleotides, and then healing up all of the little gaps in between by the DNA ligase. The factory will run, remember that the chromosome is circular. The factory will run around, but there's something to think about, and that is that in fact, at the REC, we have the possibility of running two factories. We run them in opposite directions to one another. And that is exactly what happens. Once replication begins on both sides of the REC and runs around until they meet. And when they meet, that is where termination occurs. And termination is actually quite a complicated process for the simple reason that we're looking at a double strand circle. And when we do all of our replication, at the end, we find those circles, the two new circles, are tied together like that. And again, it is the responsibility of topoisomerases to break and restructure the two circles and separate them with the help of DNA ligase, which joins up any gaps. Okay, here it all is for you, laid out, carefully laid out again. I would want you please to remember though the names of the enzymes that we discussed. Helicase, opening it up. Gyrase, relieving the tension. Remember the proteins that stabilize here. DNA, the primase that lays down the primer. Then the DNA polymerase that lays down all of the deoxyribonucleotides. And finally, then the DNA ligase, which heals up any gaps that there are, particularly on the lagging strand where we have to manufacture everything in those little fragments that must all be sewn together. So now there's just one thing to tell you, more to tell you, and that is this is actually an energetic process. Synthesizing these strands requires an input of energy. And when you have a look at the structure of a nucleotide, let's go all the way back. When you have a look at the structure of the nucleotide, just imagine this nucleotide now with 3 phosphates on it. That is the way the nucleotide normally exists in the cell. If 3 nucleotides have 3 phosphates attached to them. Wait a minute. What if this nitrogenous base here is adenine? Adenine triphosphate. Whoa. ATP. ATP is actually a nucleotide. Who knew? But it is. ATP, the most commonly used source of biologically useful energy in the cell, is itself a nucleotide. And all of the other nucleotides have the ability to function in this way, to actually carry biologically useful information in their triphosphate form. And that is exactly what they do. Those, when we add these nucleotides at the 3' hydroxyl site, these phosphate bonds are hydrolyzed, and the energy is harvested to form this double bond, the phosphodiester bond between the sugar and the phosphate. And two phosphate groups are freed from the triphosphate nucleotide. So they behave exactly the same as ATP does. The energy for forming the covalent bonds that structure the nucleic acid itself, that energy is inherent in the free nucleotide. So, okay, so here, this brilliant electron micrograph of the bacterial chromosome actually busy replicating. And if you have a look at it here, you see that there are two circles. And here is the last remnant of the original parent there. And you'll see on either side, there is a replication fork. And those replication forks are running toward, they're about to meet in the middle there. Absolutely brilliant picture. And these are the new strand, these are the new chromosomes behind. And each chromosome is composed of one parental strand and one new strand. Here are the replication forks, they're going to run together. Exactly that is shown to you here. But at the very end, the two end up being tied together just because of the geometry of replicating these double helices in a circular form. And so that there is then is actually broken and then broken. The strands are separated and they are healed by enzymes like topoisomerases and DNA ligase. And we end up with two identical bacterial chromosomes. In the plasmids, exactly the same thing, excepting that they're not attached to the REC, but they replicate in the same way. Okay, so I'm not quite sure. I hope that this works. Let's just have a look. Yes. Okay. No, hold on. Notice what's happening on the lagging strand. Okay. All right. So that takes us through the one major responsibility that DNA has, which is that it's got to act as a faithful store of the information accumulated, by the way, through evolution, through evolutionary time. And as far as possible, hand on that information entire and accurately to progeny. The second major responsibility of DNA is actually to allow those recipes that are contained in its sequence to be read by the cell when the cell requires a product which the DNA has the recipe for. So the first thing we need to understand before we start discussing the actual processes is to discuss just a little bit about this other molecule called RNA. We need to understand that the cellular mechanisms do not speak DNA. They don't speak the language of DNA. The cell cannot go directly to the DNA and just make a protein from the information contained in DNA. Instead, the cell requires an intermediary. It requires an intermediate form which it can actually interpret. And that intermediate form is RNA. RNA also, it's ribonucleic acid. Remember, it has a hydroxyl OH group at the 2' carbon. So it is a ribonucleic acid because it uses ribose. The RNA strand, unlike DNA, is not inherently double-stranded. It's single-stranded, with a caveat, which I'll explain in a little while. But it is essentially a single-stranded nucleic acid. And the third difference to DNA is, of course, that it does not use thymine. It uses a very, very closely related nucleotide, which has uracil as the nitrogenous base, not thymine. So those three differences distinguish it functionally, but they also label it for the cell to recognize. And the cell speaks RNA. It doesn't speak DNA. It speaks RNA, can talk to the RNA. And the RNA can talk to the DNA, but the cell can't talk directly to the DNA. So the next thing to know about RNA is there are actually several kinds of RNA. It's a very versatile molecule, and it has some amazing functions in the cell. We won't discuss all of them, but it has many different functions in the cell. The first is that the first kind of RNA that we're going to encounter is called messenger RNA. And messenger RNA is a transcript of the DNA code. So messenger RNA is literally that. Messenger RNA is the message that the DNA sends out to the cell to be read and interpreted. Because don't forget, the cell can't speak DNA. It can speak RNA, but it can't speak DNA. So messenger RNA, it carries all the coded information that is present in DNA in a form which the ribosomes in the cell can actually read and translate. The next kind of RNA that we're going to encounter is called transfer RNA. And transfer RNA is an amazing molecule. There are many, many kinds of transfer RNA in the cell because transfer RNA consists of a combination of RNA and an amino acid. And each amino acid has its own transfer RNAs that it attaches to. So there are at least 20 different transfer RNAs for the 20 different amino. Actually, there are more than that. But you can think for the moment, we have at least 20 transfer RNAs to fit the 20 amino acids that we use to make up proteins. Remember that amino acids are the monomers of the polypeptide polymer. The last kind of RNA that we are going to talk about is the RNA which is included in the structure of the ribosomes. And this is ribosomal RNA. And ribosomal RNA has two distinct functions. The first is it's a structural element. It actually builds the body of the ribosome. The body of the ribosome is made up of a mixture of proteins and RNA, ribosomal RNA, coiled and folded into a very, very complex structure that produces the structure of the ribosome. So that's point number one. It is structural. But number two, function number two is that some of the ribosomal RNAs are actually enzymes. We have said before that the vast majority of enzymes in the cell are actually protein, but the exception are the RNA molecules which are able to act as enzymes, and we call them the ribozymes. And some of the ribosomal RNA molecules are ribozymes, and they are intimately concerned with the process of translation, of creating the final protein molecule from the RNA code. So we're going to go through all of that. We're going to see exactly how this is done. And before we do that, we need to reintroduce you. I've talked about this before, but here is another thing that you need to be repeating as you lay your heads on the pillow at night, and that is to repeat yourself the central dogma. And the central dogma states, it identifies the way in which this process takes. It identifies the flow of genetic information in the cell. And that is that DNA is transcribed. So the DNA is transcribed to messenger RNA, to the messenger RNA. That messenger RNA strand goes out to the cytoplasm, and the messenger RNA strand is translated by the ribosome to protein. So the ribosome is actually going to produce the protein. It doesn't produce the protein really, it produces the polypeptide that will be matured to form the protein. Remember that many things may happen to this polypeptide. Remember what we said about how the cell may be processing proteins, folding them, chopping them up into different pieces, folding them, etc., putting them together. Lots of things happen to this polypeptide before it begins functioning as a protein. So this is the central dogma. DNA is transcribed to messenger RNA, which is translated to polypeptide or to protein by the ribosome. That is the central dogma. It's called the central dogma because this is almost a definition of life on the planet. This is how all living things function. This describes the process of genetic transcription for all living things. This is how living things produce their proteins and therefore produce their enzymes, and everything else results from that. All of the information for the living systems is contained in DNA, but it must be transcribed to messenger RNA. And that messenger RNA then needs to be translated into, first of all, into polypeptides, but those then go to form the functional proteins. Remember this. This is tremendously important. And it's what links us all. It's what links all living things together on the planet. These systems evolved in the prokaryotes, but they work so well that they have been preserved through evolutionary time with some modifications, as we'll see. There's some differences between prokaryotes and eukaryotes, but essentially the process itself is the same. That is why we refer to this as being the central dogma. Okay, so the very first thing that we need to know about is how this happens. How is DNA transcribed to the messenger RNA transcript? That takes place in a process which we refer to as transcription. And it's transcription at its simplest. It's the synthesis of a complementary messenger RNA strand. So somehow or other, we're going to look at the DNA code, and then we're going to transcribe it to a messenger RNA copy. And that messenger RNA copy is going to be sent out to the cell for later translation. The same rules apply. We're going to build an RNA molecule, okay? But what are we doing? We're building a nucleic acid. So we use the same rule. We can only grow it in the 5' to 3' direction. We can only add at a 3' hydroxyl group. So it obeys the same rule. It also obeys the base matching rules, with one distinct difference. When we see what is happening, we're going to open up the DNA strand and we're going to put in matching bases. We're going to expose DNA strands. We're going to put in matching bases. But wherever there's an A on the DNA, we're not going to put in a T on the RNA. Instead, we're going to put in a U, a uracil. Okay, so we're not going to see any thymine in the RNA strand. Instead, wherever there is an A on the DNA, we're going to put in a U on the RNA. So here, this is the way it happens. In some ways, you will see some similarities to the replication. First of all, there is a big complex of RNA polymerases and various other enzymes that are actually going to perform this action of transcription. In a similar way to the DNA replication factory, it's going to run down a strand and it's going to open it up, but it only opens up a little bit. Doesn't open up nearly as much as we would with a replication fork, and the opening is very temporary. The strand simply heals up behind the polymerase as it moves along, as it moves down. So first of all, we need to understand that this process is exquisitely controlled. We can't just have an RNA polymerase just wandering around the genome, latching on, and then reading off messenger RNA. There are various things that have to happen. First of all, we only read off genes when we need them. So RNA attaches to genes only when that gene needs to be read. Some genes are read off continuously, and that is for proteins that we need continuously. Those are called housekeeping genes, and they read off things like, for example, the respiratory enzymes of our respiratory chains, which need constant replacement in the membranes. Those sort of things will be read off continually, but many, many, many genes remain silent for most of the time and are only activated when the cell actually needs that particular product. How on earth do we switch these genes on and off? It's a very complicated topic. In fact, it's the subject of a whole science, basically. But I'll just tell you that every gene above the gene, we're going to watch, the RNA polymerase is going to move in on this strand in that direction, okay? And as we call this region here the upstream, and that's the downstream. So upstream of the gene here, there is a region of DNA which it's not part of the functional gene. It doesn't contain information for a protein, but it contains all the information about the gene. It's effectively, you can think of it as labeling area. And when the gene needs to be read off, the RNA polymerase identifies this region, binds to it, and then begins replication. It actually binds to it, and then it moves down to a specific marker on the DNA before it begins transcribing. So this is like a switch. And the RNA comes along, binds to that particular region when it needs to, and then begins the process of transcription. This area has a very complicated, its functioning is very complicated. We're not going to discuss it, but it's called the promoter. And it is this essential region for the control of gene activity. So that is the promoter. And we just think about it in simple terms. When the gene needs to be read, the RNA polymerases bind here at the promoter. They move down to a signal which tells them start transcription. As when transcription starts, they open up the DNA strand, they split it, and they begin laying down bases, matching RNA bases, not matching DNA bases, but matching RNA bases. In the same way as we saw with replication, they are laying them down at a 3' end. There's the 3' end of the RNA, messenger RNA strand there, that that's where they're going to add. Here's something you would probably immediately notice. And that is that in this gene, only this strand here contains the useful information. Only this strand here contains the information for the creation of the matching RNA strand. This strand here does not. So this we refer to as the template strand. So this is the template strand of the DNA. This was referred to as the non-coding. It has some way of designating that it is not going to be used. That is only true for this particular gene. Go to the next gene, and it is quite possible that this strand would be the coding strand, the template strand. Okay, so for any gene, only one of them is the template strand. The other is non-template strand. So you've got to ask yourself a question. What then is it that this strand codes for? Why have it at all? What does this strand, the non-template strand, code for? It codes for the template strand. That is how we replicate our DNA, right? Each strand codes for the matching strand. That's how we replicate our DNA. It's essential that we have this strand, the matching strand, in addition to the template strand. But it is not, this strand does not participate ever in reading of messenger RNA. Okay, so the next thing to notice, difference to replication. Here we are laying down our messenger RNA strand. We're doing it in this direction. This is the 3' end here of the messenger RNA strand. Here's the 5' end there. And you'll see that we only produce a short length at a time, which adheres to the DNA. This is a DNA RNA hybrid. And the DNA RNA hybrid is quite small. That RNA comes off the DNA, and the DNA is allowed to rewind behind this RNA polymerase as it moves down the DNA strand. The next thing to know is that you don't just have one of these RNA polymerase complexes moving down to do the transcription. We can have many, many, many of them. Once the gene is, once the promoter is activated and allows the RNA polymerase to bind, it can bind many of them very quickly. So we'd have lots of them running down the DNA, reading off messenger RNA transcripts. And we can actually see these. We can, using electron microscopy, things like that. And when we visualize them, this is the sort of thing that we see. We just see the RNA polymerase. This RNA polymerase is running down in this direction there. Here is the RNA transcript there, the messenger RNA transcript coming off. And this polymerase has been on much longer than this one. This one's been on longer than that. That one's been on longer than that one. This one's just beginning transcription. So the transcript here is much longer than it is here and here. Eventually, the RNA polymerase reaches the end of the gene. And there are various ways, which we won't discuss, where locked into the DNA sequence usually, there is a message which is given to the RNA polymerase to tell it to actually fall off, to disengage from the DNA. There is a stop instruction. It's not often not a single signal. It's also can be vary between different genes, but there is some instruction is given to the RNA polymerase to tell it to stop. And when it reaches that instruction, it falls off, and the complete RNA strand is then allowed to go free into the cytoplasm. I just, at this point, just remind you about one thing, and that is we need to think of this in two circumstances. Okay. Our main interest is in prokaryotes. In prokaryotes, this all takes place inside the cytoplasm. So this is just released off into the cytoplasm as it is. But remember that in eukaryotes, this whole process takes place inside the nucleus. And once this transcription process has ended in eukaryotes, this transcript needs to be exported. It has to be exported from the nucleus in a very complex export system, which goes through the nuclear pores and then out into the cytoplasm to be translated by the ribosomes. We'll point out some other differences in a while, but for the moment, just remember, prokaryotes, this all takes place in the cytoplasm. Eukaryotes, this process takes place inside the nucleus. Okay, let's just see. Come on. Okay, so let's leave it there because I want to have you bright and happy and everything for our discussion about translation at our next class. Okay, so with that, I bid you good day, and I hope you are all well. Marks for that midterm were really, really very good. I was very, very pleased. You guys must be working very hard. I know that it's not always easy, but I was really, really quite pleased with the results of the midterm. And from here, we go onward and upward.